Hello, my name is Dr. Eva Sinapidou, and I'm going to talk to you about local plant varieties as a genetic pool, their maintenance through traditional techniques and modern tools. I am an assistant professor in biotechnology of plant protection, and my expertise is in molecular plant pathology. I have been working in the Department of Agricultural Development, Democritus University of Thrace, since 2010. My main research involves plant breeding in combination with molecular interactions between plants and microorganisms, both symbiotic and pathogenic to plants. Let's get familiar with some terms. A species is often defined as the largest group of organisms in which any two individuals of the appropriate mating types can produce fertile offspring, typically by sexual reproduction. The term species is a familiar unit of botanical classification within the plant kingdom. It is clear that within a species, there can be a wide range of different types of plants. Farmers and growers need plants with particular characteristics that they are adapted to their environment and their cultivation practices. A variety represents a more precisely defined group of plants within a species with a common set of characteristics. In the classification system of plant taxonomy, variety falls below species in taxonomic rank. A variety refers to a variation within a plant species that develops naturally in the environment. Unlike cultivated plants, a variety does not require human intervention to grow and reproduce. Seeds from a specific variety will often grow true to type, meaning that their offspring will retain the parent plant unique characteristics. A plant cultivar refers to a variation within a plant species that has been developed by a human through controlled plant breeding, as opposed to occurring naturally. The term cultivar means cultivated variety. To create a new cultivar, growers crossbreed parent plants that each have desirable characteristics in the hope that their offspring will inherit those traits. Once a grower ends up with a new cultivar with the desired traits, the grower must then ensure future generations of that cultivar maintain the distinguished characteristics. To accomplish this, the grower has to use either controlled pollination or various methods of asexual propagation. There are guidelines for how to write a variety name. When writing a variety name within a plant's full scientific name, the species name comes first, followed by the abbreviation var, followed by the variety name, all in lowercase and italicized, as shown in the example. When writing the complete scientific name of a plant cultivar, the cultivar name occurs after the name of the genus and species. The cultivar name is unitalicized, set off in single quotation marks, and the first letter of each word is capitalized, as shown in the example. The term plant variety has a different meaning. A plant variety is a legal term coined by the International Union for the Protection of New Varieties of Plants that recognizes certain cultivars as official plant varieties in order to grant the creator of the cultivar legal protection, commonly referred to as plant breeders' rights. Land races are populations of plants that have been cultivated for many generations in a certain region, being shaped by biotic and abiotic stresses, crop management, seed handling, and eating preferences. In this sense, a land race is a dynamic population of a cultivated plant that has historical origin, distinct identity, and lacks formal crop improvement, as well as often being genetically diverse, locally adapted, and associated with traditional farming system. Land races can be found in all cultivated plant species, crops, vines, fruit trees, vegetable, and ornamental plants. They are a part of local culture. Due to their special characteristics, they can play an important role in local economies. Most frequently, they are not adapted for mass production. So the major characteristics of land races are high levels of genetic diversity within populations, adaptation to soil and climate conditions typical of the region combined with resistance to common pests, edible parts that are valued by local people, normally shaping and being shaped by the local cuisine, and modest but stable yield, conferring food security to the local community under normal environmental variation. Due to their wealth in genetic variability and adaptability to different environments, land races are the most valuable genetic resource for long-term plant breeding programs and also prime target for germplasm collections. Seed banks around the world keep thousands of samples of land races. In some countries, there are efforts to devise regulatory mechanisms and financial incentives for traditional communities to keep growing their heritage varieties. New systems for germplasm conservation have been built on social networks connecting people interested in the subject. Those networks take advantage of modern communication tools to replicate on a global scale what used to happen through personal contact in traditional communities. In self-pollinating species such as rice and wheat, land races can be thought of as a mixture of pure lines, including some heterozygous individuals derived from a low frequency of cross-pollination. In this type of population, selecting single plants and deriving inbred progenies invariably result in some lines that outperform the original land race for a given growing condition. However, this superiority comes at a cost. Because pure lines are normally less stable than diverse populations in the face of stress, especially diseases, and have no capacity for long-term adaptation because it is monomorphic for most genes. In the case of open pollinated species, such as maize, land races are populations of random mating individuals. Mass selection and recombination of the top performing portion of the population result in a gradual increase in the frequency of favorable alleles. Successive generations of selection on maize landraces resulted in improved open pollinated varieties, which were the basis of corn production until the advent of hybrid maize. Plant breeding is an ancient activity dating to the very beginnings of agriculture. Probably soon after the earliest domestication of cereal grains, humans began to recognize degrees of excellence among the plants in their fields and saved seed from the best for planting new crops. Scientific plant breeding dates back hardly more than 50 years. The role of pollination and fertilization in the process of reproduction was not widely appreciated even 100 years ago. And it was not until the early part of the 20th century that the laws of genetic inheritance were recognized, and the beginning was made towards applying them to the improvement of plants. Plant breeding started with plant domestication that is responsible for the origin of crops and evolved through the intuitive farmer selection that led to landraces and ended up to pure line selection and mass selection that produced plant cultivars. The plant breeder usually has in mind an ideal plant that combines a maximum number of desirable characteristics. These may include resistance to diseases and insects, tolerance to heat and frost, appropriate size, shape, and time to maturity, and many other general and specific traits that contribute to improved adaptation to the environment, ease in growing and handling, greater yield, and better quality. Thus, the breeder can rarely focus his attention on any one characteristic, but must take into account the manifold traits that make the plant more useful in fulfilling the purpose for which it is grown. Plant breeders can be gardeners, farmers, experts, companies, universities, and research institutes. The vast diversity of breeding methods can be simplified into three categories: plant breeding based on observed variation by selection of plants based on natural variants appearing in nature or within traditional varieties; plant breeding based on controlled mating by selection of plants presenting recombination of desirable genes from different parents; and plant breeding based on monitored recombination by selection of specific genes or marker profiles using molecular tools for tracking within genome variation. Despite the great spontaneous diversity that can be found in the landraces, simply applying selection on pre-existing diversity is an eroding process that eventually comes to a limit. The true creative power of plant breeding resides in promoting recombination for favorable alleles. The combination of different alleles in many loci results in virtually infinite number of genotypes. Given the myriad of possible genotypes resulting from crossing different parents, the limitation for genetic gain becomes the capacity of a breeding program to evaluate large number of plants deriving from a number of crosses. Pedigree breeding consists of crossing parents and generating segregating populations, which are conducted through generations of self-pollination and selection until a set of derived lines that combines the good characteristics of both parents is obtained. The pedigree method is appealing to breeders because it allows building better varieties by putting together in the same plant good characteristics that were present in different materials. This method is efficient for breeding for qualitative traits, such as disease resistance, or easily classifiable traits, such as plant architecture, or the color or shape of plant parts. The ideotype breeding can be regarded as a strategy to improve the capacity of the pedigree method to promote gain for quantitative traits such as yield. Changing the ideotype of a crop often requires looking for variation beyond the boundaries of elite germplasm, which is normally of the current plant type. In modern population breeding, the objective is to increase the value of the population as a source of elite lines. Improving the mean quality of the population while preserving the variation within it results in top individuals that outperform previously existing lines. Those lines can be used as cultivars in the case of self-pollinating species, or as parents of hybrids in the case of cross-pollinating species. Traditional breeding methods were based on the complementarity between parental characteristics. However, little or nothing was known about which part of the genome came from each parent. This situation changed with the advent and dissemination of molecular marker techniques, which made it possible to monitor the transmission of chromosome segments in the progeny. When many molecular markers are genotyped in a set of plants that have derived from a single cross, the frequency of recombination between them can be used to infer their order and relative distance in the chromosomes, resulting in what is known as a genetic map. From a QTL map, a plant breeder can get an insight into the number of loci controlling a trait, their relative importance, and their approximate position in the genome. Whereas in the case of marker-assisted gene introgression, the breeder will normally tag only one gene, a full-feature marker-assisted selection scheme would monitor several genes simultaneously. Knowledge of the allele borne by each plant in each major locus of interest creates the means for building specific allele combinations that would maximize the agronomic value of the line. The genomic selection approach proposes that knowledge of the relationship between specific markers and specific genes is not necessary in the breeding context. Instead, the breeder can use the information available for all markers in a plant to predict its breeding value without evaluating its phenotype based on previous statistical models built for those markers using a training population for which all genotypes and phenotypes have been scored. The raw material of natural genetic diversity was carved by human selection during plant domestication resulting in profound changes in plant phenotypes. Intentional or unintentional selection through millennia of traditional farming resulted in a wealth of genetic diversity adapted to different human needs. However, this process worked at a speed that no longer matches the demands of modern society. The most modern methods will be necessary for going back to what remains from the raw material where our ancestors found their sources of food to find the genes that will be building blocks of the cultivars that will solve the problems of the 21st century. Plant breeding can be considered a co-evolutionary process between humans and edible plants. People caused changes in the plants that were used for agriculture, and in turn, those new plant types allowed changes in human populations to take place. From this point of view, it becomes clear that plant breeding is one of the main foundations of human civilization. These are recommended articles of the topic covered. Thank you for your attention.